Hi class, welcome to our first official lecture of our online portion of this class. So if you've looked, this is also the first lecture of our third unit. So if you look at this, if you've taken a look at the chapters in this unit, you probably already realize that this unit is all about genetics and DNA and gene expression and how that actually works in bacteria. So what we're going to do in this first chapter, this first lecture, lecture number 8, is talk about the molecular biology behind. Why isn't my slide advancing here? Let's double check. Okay, there we go. It always takes a few clicks initially. I don't know why. Well, first of all, I want to talk about a couple things. One, I apologize for getting this lecture up so late. I was having a really hard time figuring out. Well, one, I had to do a ton of revisions. This is a very tricky content to teach online because there's a lot of moving parts. And so I finally, I think, figured out a way to show video while I'm talking to you via this interface that I'm using. And then the other issue was that when I was using the videos, it wasn't uploading to YouTube. I don't know if they were taking it down because it was a content, like a copyright issue, or if the video is too long or what it is, but I'm trying to make the videos a little bit smaller so that we don't have that issue going forward. So even though this is all one lecture, lecture number 8, I'm probably going to divide it into at least 3, maybe even 4 videos and put those on Canvas. So hopefully I've ironed out the kinks here and we can make sure this works going forward. So even though this is lecture 8, it's focused mostly on chapter 7 of the Nestor text. You can see the majority of the sections are from chapter 7. There's one section on mutations from chapter 8 that we'll be using as well. So before we get into how DNA and RNA works, do a little bit of a review from 107, I want to very briefly talk about the history behind how DNA was discovered, just because it's such an interesting, I'm kind of a history buff and I really enjoy thinking about the way that these experiments kind of led to the elucidation of DNA as a hereditary molecule. People don't realize that we only learned about this less than 100 years ago, about 80, up until through the 30s, I would say through the 1930s, most people still thought that protein was the hereditary molecule that passed information from one generation to the next. Everyone knew on some level about heredity, and they knew that it passed through a series of how would I say, there were rules that applied, right? Think about Mendelian genetics, right? The dominance and recessive traits. And there had to be some mechanism for that information flow. But scientists didn't know what that molecule was. And most scientists, again, thought it was protein, but they didn't understand how it worked or the mechanism of that passage of information. They did know, however, that whatever the molecule was, whether it was a protein, a lipid, a carb, or a nucleic acid, they did know that it had to be stable. So this molecule had to have some stability to it in order to be passed along, but at the same time, it had to have some mechanism for change because they knew about mutations by this time, and they knew that there was some type of, there had to be some mechanism in the molecule that introduced changes or mutations in that molecule while staying stable at the same time. That was kind of those two things seem incongruous until you learn about the actual structure of the nucleic acid molecules themselves. And then finally, the molecule itself had to be able to be replicated in some way. There had to be some neat mechanism for replication. And again, scientists just thought that protein served as the basis for their own replication. There was like a kind of like the protein itself was a mold for making another protein, and that's potentially how it worked. So there was a lot of controversy surrounding this idea, not only in what the molecule was, but how the molecule worked and how it was passed on, how it was replicated, and so on. So through most of the 30s, by the 40s, there was a couple of interesting experiments that came around. There was an experiment by, I think his first name is Thomas. They're called the Avery experiments. We'll talk about them in the next section. But there was another series of experiments by Hershey Chase, by doctors Hershey and Chase. Then they have therefore, the very famous Hershey-Chase experiment is named after them. It's also called, I think it's funny, I had to include this, it's also called the Waring blender experiment because they used, I don't know if you know what a Waring blender is. It's like it's the blender that like bartenders use. To this day, it's a brand of kitchen blender that is used, and so they use this blender in their experiment. So it's colloquially known among scientists as the Waring blender experiment. And so what they did was a very elegant experiment, really. What they did, so at the time they knew about bacteriophages. They knew that there were these viruses that could infect bacteria, and so they took these viruses and they either in this top row, they took these viruses and they labeled the proteins. They radioactively labeled the proteins. So in this virus, in the bacteriophage, the proteins were all radioactive. Now, in a separate experiment, they did basically the same thing, but instead of labeling the proteins, they labeled the DNA. So now in this bottom experiment, the DNA is radioactive. So they allowed these bacteriophages to infect bacteria. So they grew the bacteria in these flasks. And then they took the bacteria, they put them in a blender, and then they turned the blender on. And what that did is just the torsional force, right, the centrifugal force of the blender ripped the bacteriophages off of the surface of these bacteria. And then they analyzed and they spun, so then they took the material that was in the blender and spun it down so that the bacteria were on the bottom and all the bacteriophages were on the top. And what they found is when they radioactively labeled the protein, the radioactivity was found in the supernatant. In other words, the proteins stayed with the bacteriophages. So the bacteriophage, the shell, the nucleocapsid, the tail, the spikes, right? All of those things that we know make up bacteriophages were radioactively labeled, and they did not get into the cell. And as we know, if we look at this bottom one, what actually gets into the cell, what gets into the bacteria, of course, is the DNA. So when they radio-labeled the DNA and ran it through the same experiment, right, blended it, spun it down, they saw that the radioactivity was actually in the bacteria here instead of in the bacteriophages. So this experiment was really it was very well recognized and accepted at the time. In fact, I think Hershey, he won, I want to say maybe in the early 50s, maybe 1950, 1951, he won the Nobel Prize, for this experiment. And, his colleague, Martha Chase, a woman, not only didn't win, not only didn't share the Nobel Prize for it, but was not acknowledged in any way for her contributions to this experiment. So, this experiment was really one of the first experiments to conclusively show that DNA is what gets into the nucleic acids are what get into the bacteria when a bacteriophage infects a bacteria and takes over and starts making that bacteria process new instructions. So this was a very good hint that DNA was the molecule of inheritance, or the molecule that contained information for bacteria. Even though they had there was this big advance, right? And they showed there was other advances. Again, the Avery experiments, there's a nice, let me look in the book. There's, in the intro portion of our book, in the very first section, I forget the name, the group that did this, they won a Nobel Prize too. Let me see. Beadle and Tatum. Beadle and Tatum mutated some type of bread spore, I want to say. It was like some type of fungus, and they saw that the loss of enzymatic function was tied to what they called a gene. But at the time, again, we can call it a gene, we can call it a molecule of inheritance, we knew that there was DNA and RNA in cells, and all cells, but we still didn't understand how it all came together. In other words, how does this molecule actually work? What do the instructions look like? How is it replicated? How can it be stable? And so on. And it wasn't until the early 50s that the final piece of the puzzle kind of clicked into place with the elucidation of the actual structure of DNA. And of course, this was very famously done by a couple of researchers at Cambridge University in Britain, Francis Crick and James Watson. Again, what many people don't fail to remember is that they, first of all, Maurice Wilkins shared the Nobel Prize with them. And all of this was based on this very, very famous picture called Picture 51 or Photo 51. That is an X-ray crystallography picture of the molecule DNA. And this is what allowed Francis and Crick to figure out the structure of DNA. And this was Rosalind Franklin's work. And they actually stole it from her. They took it from her lab without her permission or her knowledge. And basically worked with Maurice Wilkins to steal her work, and they got a Nobel Prize for it, and she was never acknowledged, and she passed away from ovarian cancer several years later. So it's a tough time to be a woman in molecular biology. Clearly the 40s and 50s weren't kind to female scientists. So I'll get off my soapbox here and go back to biology. so Francis and Crick, again, credited with figuring out the structure of DNA. So let's talk about what that structure actually is. Remember that DNA is nucleic acids are a type of macromolecule. We have 4 major types. We have lipids, carbs, proteins, and nucleic acids. And what I kind of like to think about is when we think about lipids and carbs and proteins, there are thousands, if not millions, of different kinds, right? There's all different kinds of carbs. Think about just all the different sugars that you could probably name a half dozen different sugars on your own, right? Think about all the different proteins, all the different types of fats and waxes and oils there are. Nucleic acids have 2 molecules. That's it. DNA and RNA. And the monomer that makes up DNA and RNA is called a nucleotide. So most macromolecules have a single subunit that has some form of a repeat that makes up that macromolecule. And for nucleic acids, that monomer is called a nucleotide. So let's look at the structure of a nucleotide. A nucleotide has 3 smaller subunits that make up this nucleotide. And believe it or not, DNA is actually made up. Its base, its kind of central subunit, its central portion is actually, is actually a sugar. It's a ribose, so it's a 5-carbon sugar. And we call it a deoxyribose because of its structure. Let's talk about this sugar in a little bit more detail so you understand when I'm using these terms later. When we're talking about organic chemistry, in other words, carbon and hydrogen-based chemistry, it's common to number the carbons in a molecule. So you see these blue numbers here, carbon number 1. So there's a carbon at this little corner right here. The second carbon is here. The third carbon is here. Carbon number 4. And then finally, carbon number 5 is up here. So a ribose sugar is a 5-carbon sugar. And you can see here that at that second carbon, there's no hydroxyl group here. So we're going to call this particular kind of ribose a deoxyribose. It's lacking in oxygen here at carbon number 2. You can see that at carbon number 1, we have this sugar is attached to a nitrogenous base they're also called nucleobases. I tend to call them nitrogenous bases. And on the 5 prime carbon and the 5th carbon on this sugar, we have what's called a phosphate group attached to that 5th carbon on our sugar chain. So this is our monomer. This is our nucleotide for DNA and specifically, let's just talk about DNA for now. And one of the things that I like to point out about DNA and RNA as well is that it has a variable portion and a constant portion. So these 2 things, the ribose sugar and the phosphate group, will stay constant. In other words, they never change. We'll always have this phosphate group, and we'll always have this sugar attached to each other in this way and attached to a nitrogenous base. However, this nitrogenous base is variable. And this is starting to hint at a couple of those requirements for our molecule of heredity. Remember, it not only has to be stable, but it also has to have a mechanism for variation. So this sugar phosphate gives it stability. It contributes to its stability. It's one reason why it's stable is that it has this very, very stable, very highly negatively charged sugar phosphate backbone. But then it can also have variability in that its nitrogenous bases can change. So let's talk about these nitrogenous bases. There's a lot of long words in this lecture. Our nitrogenous bases can be categorized into 2 different types, purines and pyrimidines. Purines have a double ring, and they consist of adenines and guanines. So this particular nucleotide is a purine, and we know this because it has adenine as a nitrogenous base. Now, adenine and guanine are found both in DNA and RNA. But let's look at our pyrimidines because there is some variation here. Pyrimidines not only differ from purines in that they have a single ring. You can see the structure of these molecules have a single ring structure. But what's interesting about pyrimidines is that thymine and uracil are kind of complementary. Well, that's not really the word. They're what would be a word to describe these? Well, so thymine only exists in DNA. So if we see a nucleic acid and it only has it has thymine in it, it has T in it, then or T in it, that sounded odd. If it has thymine, this nitrogenous base in it, we know that nucleic acid is DNA. Whereas if we look at some nucleic acid and it has uracil in it, we know that nucleic acid has to be RNA. So they're kind of like oh my goodness, I can't think of the word. They're corollaries. They're corollaries to one another. So thymine is a pyrimidine that will always be in DNA, whereas uracil is the nitrogenous base or the nucleotide that will only ever be in RNA. Further, we can further characterize these based on how they pair up with one another. And I'll talk about that, I think, in the next slide. Great. Yep. So let's go a little bit further into the structure of DNA. I mentioned in the previous slide about a sugar phosphate backbone. So let's look at this first. Let's look at the red, kind of the red highlighted side of this DNA. Remember that DNA, I'm sure you remember, it's called the way that we talk about DNA is a double helix. It's a spiral shape that looks almost like a ladder that's been twisted. And the rungs of the ladder, you can see we've colored the outside of the DNA strands red and blue. So the red strand, let's look at the red strand here first. So the sides of the ladder, kind of the part that you would like grab onto with your hands as you're walking up the ladder. Another way to describe that is the sugar phosphate backbone. And you can see here that we basically have these alternating sugar, phosphate, and so on. And if you think about DNA, remember DNA doesn't exist on paper. It's not two-dimensional, it's three-dimensional. It's in our cells and it can be twisted every which way. So we can't really refer to DNA as a left side and a right side because it can just flip upside down and then it doesn't work. There's no top and bottom, there's no left and right and so on. The way that we refer to directions in DNA and I guess RNA as well is by the end of the carbon. Remember how important I talked about numbering those carbons. If we were to look at our ribose sugar here, this kind of carbon hanging out here is our third carbon. Remember carbon number 1, carbon number 2, and here's our third carbon that has that hydroxyl group hanging off the end of it. And if we were to continue up this sugar phosphate backbone, we'll see that this one will end in a phosphate. A way to describe this phosphate is a 5' phosphate because it's connected to that 5th carbon on our chain. So the directionality in DNA has basically, we refer to one end as the 3' end. So I usually call it the 3' sugar because it ends with sugar. Some people refer to it as a 3' hydroxyl. Both are fine. And then on the other end, we're going to have a 5' phosphate. So instead of saying bottom and top, for example, we could refer to this as the bottom and this as the top. What we really should do is say this is the 3' end and this is the 5' end. And the reason that we can't refer to this as the bottom is because what's bottom for one is the top for another, because DNA is what's called anti-parallel. It's kind of like a two-way street where the streets will never converge with one another. They run alongside one another, but the cars are moving in opposite directions. So when we think about this, the red strand is going from the 3' here on one end to the 5' at the other. The blue strand is going to be running in the opposite direction. So here, where we have the 5' end on the red strand, we're going to have the 3' end on the other strand. So again, we still have our sugar phosphate backbone, but notice it's essentially upside down, right? If we were to flip this picture, it would still be the same orientation. It's just that now, and this is why we can't use that top and bottom or left and right nomenclature when we talk about DNA. So we have the 3' end here at the top, for now at the top, because we're on paper, it's a stable picture. 3' end here, and then we have the 5' end, the 5' phosphate at this end here. Notice it'll always be opposite of its complementary strand. So now let's talk about our pyrimidines and our purines, how they pair up with one another. Remember, this will always, our sugar phosphate backbone will always stay the same. And our nucleotides are what is going to be different. And our nucleotides, remember, we have our purines A and G, and they're always going to pair up with their complementary pyrimidine, which is, of course, T and C. Notice, so in other words, if we know this is A, we don't even have to see that this is T. We know it has to be T. It can't be anything else. So A always has to pair up with T, and G, guanine, always has to pair up with cytosine. The other thing that I want to point out is that these are held together, our nitrogenous bases aren't held together by proper bonds. They're not covalently or ionically attached to one another. They're actually attracted to one another through hydrogen bonding, which, as you kind of remember from chemistry, hydrogen bonding is a very weak form of a bond. It's not even a real bond. It's really just a magnetic attraction between two disparate charges. And when adenine and thymine pair up, they're going to have two hydrogen bonds exist between them. However, when C and G pair together, they're going to have three. So this is a stronger attractive force when compared to adenine and thymine. And what's fascinating about this is showing us, this is another hint about how DNA works. Because DNA is held together by these very weak hydrogen bonds, and again, I'm not sure how your chemistry teacher described it to you, but I've always thought of hydrogen bonds like kind of like Velcro. They come together quite easily, but they also come apart quite easily as well. And this shows us how DNA can form as a template for its own replication, right? If we can pull these kind of two sides of a zipper apart, or two sides of a ladder apart from one another, we can then see the complementary sequences and replicate them quite easily, and then they can go right back together. So those hydrogen bonds here really form. Can you guys even see me? I'm using my hands a lot. Oh, I didn't even think of that. I'm sorry. Let me see. I don't know if that helps. Okay. So these complementary strands, even though they're anti-parallel, they're complementary to one another, and they really work very much like Velcro. And I think, when did I talk about this? I'll talk about the stability here in a bit when I'm comparing it to RNA. Okay. So before we get into RNA, let's talk about a DNA-specific process, and that process, of course, is replication. So when we use DNA to serve as a template for replicating more DNA, RNA isn't really involved, except for one very specific part, which I'll talk about. But DNA replication is really the making of DNA, using DNA as a template for its own replication. So I'm not going to spend too much time on this particular slide, just because it kind of goes through all the different enzymes that are used. And I don't want to, let me see if I can, I feel like this isn't, my screen might be cutting off. There we go. It might be slightly cutting off the bottom of my slide. So I just fixed it there, hopefully. Then can I move this as well? Oh, I can. Excellent. Good. Okay. So I'm not going to spend too much time on the various enzymes. I'll probably end up talking about them as I'm describing these mechanisms, but I do want to point out DNA polymerase 3 because this is the big one. This is the enzyme that really does the majority of the work, even though this is a team effort. DNA polymerase 3 is the enzyme that really makes this happen, and it also has some characteristics that necessitate some complications in DNA replication. The first complication is that DNA polymerase 3 only works in one direction. In other words, it can only add nucleotides on the 3' end of a already existing chain. So it can't just make new DNA from scratch. It can't just start stitching nucleotides together at random. It has to add the nucleotides one at a time. And it can only add to that 3' end of a growing chain, and it has to have a chain already started for it to work. There's a couple cool things I think about DNA polymerase 3. And one, it's crazy fast. It works. I have it. 50,000 base pairs. So BP stands for base pairs. So the A's, T's, C's, and G's, the nucleotides themselves. It can add 50,000 a minute at optimum growth conditions. Like if we're talking about a mesophilic bacteria, E. coli, let's say we were giving E. coli everything it needs to replicate, plenty of energy, plenty of nutrients, and so on. that DNA polymerase can fly. And not only can it work really, really fast, it actually proofreads itself. So it has its own like little spell checker, which I think is amazing. You know, think about trying to type as fast as you can, right? You'd never be as fast as DNA polymerase 3, and you're not checking, you know, you have to have a computer do spell check for you. This polymerase not only is super fast, but it edits itself as it's working, which I think is really, really cool. So it has like a proofreading capability. Okay, so when we talk about DNA replication, the model that is commonly, the name of the model that we use to describe DNA replication is called a semi-conservative model. And the reason that it's called semi, right, means basically half or partially. It's partially conserved. So in other words, we conserve part of this DNA strand. If you recall, let's briefly talk about the way that bacteria replicate. They replicate through, or I shouldn't say, reproduce, right? The way that bacteria reproduces through a simple process called binary fission. Essentially, one bacteria will divide into two. And in order for that to happen, it has to replicate its chromosome. Remember that the chromosome in bacteria is very simple. It has one circular piece of DNA. And that piece of DNA, that circular piece of DNA, has to make a copy of itself. And that copy is semi-conserved. In other words, when we make a copy, half of that new copy is going to be the original parent DNA, and the other half, the other strand, is going to be newly synthesized DNA. So this model is called, it was another very famous experiment by Maselson and Stahl. Maselson actually gave my class a lecture actually when I was in grad school. He's a very cool guy. But they also won the Nobel Prize, I think in the 50s, for this very elegant experiment that showed that DNA was half conserved by every passage of bacteria. So I'm gonna actually go to, I really hope this works. I'm gonna go to our online learning platform that I have access to and show you a very quick video on how this works. Let me make sure that the closed captioning is on.
The genetic information in a bacterial cell is stored in the form of a double-stranded, covalently closed circle of DNA. Replication begins at a specific site called the origin. The origin replicates, and then DNA replication proceeds in two directions. The two original strands, shown as solid lines, serve as the templates for synthesis of new strands, shown as dotted lines. This is referred to as semi-conservative replication.
Great. So again, I don't know why, I feel like I'm kind of cheating by showing videos during my lecture. But these processes are so complex that it's impossible to try to explain these on paper. And I feel like trying to is just going to make you more confused. So I'm going to pause my lecture and show you videos throughout this initial lecture. So again, semi-conservative, we can see this really beautiful picture, right? This is a real life, I think this is a scanning electron microscope image of that replication fork. And again, this DNA replication is initiated at a very specific site called the origin of replication. And the replication forks proceed in both directions until they get to the bottom where we see a termination site, and we finally have these two new pieces, two new chromosomes, two new bacterial chromosomes, each one containing one old strand, one original strand, and one new strand. So when we talk about these processes, so really this first lecture, I'm going to talk about three processes: replication, transcription, and translation. And all three of these processes have essentially three stages. Initiation, so a very specific set of circumstances lead to initiation. Then elongation, how does it proceed and how does it end? So we have three stages: initiation, elongation, and termination. So let's talk about how initiation and elongation begin. Remember that initiation is going to start at a specific sequence of DNA called the origin of replication, or the replication origin. And this site basically starts to recruit, or the site is able to recruit specific enzymes that will start the replication process. So once those two strands of DNA come apart at the origin, all of the machinery, all the enzymatic machinery can come in and start adding new nucleotides to that new parent strand. And you can see that happening here. We have DNA polymerase 3, it's this orange blob, and it's going to start adding nucleotides. Remember, it's going to add to that 3' end. So here we can see that the next nucleotide on our parent strand is T. So this enzyme, this DNA polymerase enzyme, is going to add an A nucleotide because A is complementary to T. It's going to add that A nucleotide to the 3' end of the new strand. So our bottom strand, this blue strand, is our new strand of DNA. Remember that there are some limitations. Why is this so? Let me see if I can move this down again. There we go. What are the limitations? Let's remind ourselves there's some limitations for DNA polymerase. Namely, it has to have a primer. It can only add nucleotides to an existing strand. And the other drawback is that it can only add nucleotides in a 3' direction. So it can only add it to that hydroxyl end of a growing DNA strand. So the implications for that, of course, let me go to this next strand. If we look at DNA replication and how it actually proceeds, so if with the DNA is unwinding, right, our enzyme, the DNA gyrase, is unwinding our DNA in this direction towards the left, and we started up here, our origin of replication is somewhere over here on the right, our top strand is going to be great. It's going to be just fine. It's going to start over here, our new strand, and it's going to elongate in the 3' direction. So we're adding, right, our DNA polymerase 3 is adding nucleotides to the 3' end of the new strand. And again, this just happens to be the top strand here. It could be either top or bottom. But because our template strand is going from a 3' to 5', our new strand is the opposite. Remember, it's always anti-parallel. So our new strand is running in a 5' to 3' direction. And so it can follow right along behind our DNA helicase, that big enzyme that's unzipping our DNA, and it can chug along right behind it and add new nucleotides. This is great. This is what we want if only life were that simple, right? Let's look at the bottom strand. Our bottom strand, however, is anti-parallel to our top strand, right? And this means that it's running in a 5' to 3' direction. So that means that our new strand on the bottom, and again, in this case, it just happens to be on the bottom, is going to be running in a 3' to 5' direction. And therefore, nucleotides, if it's following this helicase, it would be adding nucleotides to the 5' end of this strand. This is no good. This can't happen. Remember that DNA polymerase 3 can't do this, so we know that this can't happen. And for a very long time after the structure, the molecular structure of DNA was discovered, scientists could not figure out how this bottom strand, so the nomenclature that we use now, this top strand is called the leading strand, and scientists could not figure out how the lagging strand, so this strand that doesn't follow the way it's supposed to, scientists didn't understand how it could be replicated. They knew that DNA polymerase 3 can only add to the 3' end, so they didn't understand how it worked at the replication fork. So the way that they figured it out is that the leading strand actually undergoes a type of replication that's called continuous. So in other words, nucleotides are added in a continuous fashion as the DNA unzips. Remember, DNA gyrase is right, or no, DNA helicase, I'm sorry. DNA helicase is right here, unwinding the DNA. And so the top strand, the leading strand, comes along great. Again, if only life were this simple, we would have this really nice continuous strand of DNA being synthesized from our template strand. However, what happens on the lagging strand is a method of replication called discontinuous replication. And we see this because these very specific, I think they're about 1,000 to 2,000 base pair long fragments of DNA appear on our lagging strand, and we call these these little fragments Okazaki fragments after the scientist that is credited, again, another Nobel Prize. So lots of Nobel Prizes in molecular biology in the last century. It was named after the scientist who figured this discontinuous strand replication out, a scientist named Dr. Okazaki. So what happens here is DNA is added in a discontinuous fashion, and essentially it keeps backing up as the strand unwinds. Let's talk about this a little bit more, and then I'll show you a video. This is, again, a video will really help understand lagging strand synthesis. Again, leading strand, great, right? It comes along, adds in that 3'. The leading strand is being synthesized in a 5' to 3' direction. It's following right alongside that helicase, but again, that lagging strand is discontinuous. It can't occur in the same fashion that that leading strand can. So let's watch a quick video. Whoops. No, that's fine. Well, let's watch a quick video on how lagging strand synthesis occurs. Bidirectional, the replication fork.
DNA replication begins when the enzyme helicase unwinds a segment of the DNA and opens up the hydrogen bonding between the two complementary strands of DNA. DNA polymerase, the enzyme that synthesizes DNA, can add new nucleotides only to the free 3' end of a nucleotide strand. To provide a free 3' starting point, a short primer is synthesized first. Synthesis of the other strand of DNA is more complex. To provide a free 3' starting point on this strand, several short primers must be added. DNA polymerase then adds nucleotides to the 3' end of the primers. Finally, the enzyme, DNA ligase, forms a bond between the short DNA strands. During DNA replication, the leading and lagging strands are continuously synthesized.
Okay, so, this paused it in a really nice, no. Let me go back here really quick.
DNA replication begins when the enzyme helicase unwinds a segment of...
Right there. Okay, don't move. Is it gonna...
DNA replication begins...
I'm trying to pause it on the picture. Right there. Okay, great. So again, leading strand, super simple. Our primase, that enzyme, comes in, starts that strand off with a little piece of RNA, and then DNA polymerase 3, the orange enzyme here, just takes off and keeps adding those nucleotides in a continuous fashion on the leading strand. The lagging strand, of course, is a lot more complicated because it can't go in a continuous fashion. It basically has to keep leapfrogging over these RNA primers to make these DNA segments. So again, we have multiple RNA primers on the template strand that's put there by primase. DNA polymerase comes in, we'll add those we'll add the nucleotides again in a 5' to 3' direction. They're synthesized in a 5' to 3' direction. DNA polymerase 1, again, another type of enzyme, will come in and remove those RNA primers and put DNA nucleotides in. Remember, RNA doesn't belong in DNA. So we're going to remove those RNA, the green bits of RNA. DNA polymerase 1 takes those out and puts DNA bits in. And then finally, DNA ligase comes in and stitches all of these pieces together. So lots of enzymes, very complicated on that lagging strand, and also lots and lots of energy. DNA replication is an incredibly energy intense process, even on the leading strand, even when it's simple. There's lots of unwinding. This helicase takes up a lot of energy. There's another enzyme that's further upstream called gyrase that helps unwind the DNA even further. And all of these enzymes require lots and lots of ATP. Okay, let's go back to my lecture here really quick. How am I doing on time? I want to just look. Okay, I'm nearing 40 minutes. I'll probably finish up replication here and then stop and do a new lecture. So finally, again, we have initiation at the origin of replication, where DNA replication is initiated, and we have elongation occurring at the replication forks, and then and finally replication is going to stop at a very particular, a very specific site called the termination site, or the TER site, and it's usually on the opposite side of the chromosome than the site of origin. Here's our origin of replication, and then here's our site of termination. And you can see that from the origin, right, we have our two replication forks proceeding around the circle in a bidirectional fashion, and it's going to end at that termination site, at which point we now have two new strands on our two original strands. So we have now two complete chromosomes that will go one in each daughter cell. Okay, I think that's a good place to end it here. I'm already I talked too much. I'm already at 40 minutes. I hope that, let's hope this works. This is like my 5th try of a video, and I really want this to upload. So next up will be, we'll talk about RNA and transcription and translation. Uh-oh, did I get it? Pause. Am I pausing? I'm not pausing. Wait a minute. I don't know why this is so bad. I know you guys are laughing at me at home.